Good afternoon, my dear students. In today's live class, we had discussed about vegetative propagation, and we know that in some plants like rose, sugarcane, grapes, etc., the plant parts like stems, roots, and leaves can be used for producing a new plant. That method is called as vegetative propagation. The vegetative parts of plants like roots, stems and leaves can be used for raising a new individual and under suitable condition. And in that kind of propagation, only plants are selected. Animals have no animals are not selected for vegetative propagation. Only plants have this ability of propagating by vegetative parts. Okay, and this vegetative propagation include different methods like budding, layering, grafting that can be done in different plants like sugarcane, grapes, then roses, etc. And the most important advantages of this vegetative propagation are earlier production of fruits and flowers. We know that normally the plants are raising from their seeds, right? So in the case of this vegetative propagation, other plant parts like roots, stems, then leaves are used for the production of a new plant. Okay, so in this case, like in vegetative propagation, the fruits and flower formation will be much earlier than that of the plants that are raised from seeds. These vegetatively propagated plants have the ability to produce fruits and flowers in a earlier stage. That is the first advantage. And the second advantage is the plant that is produced by vegetative propagation will be genetically similar to their parent. So the characteristics of all characteristics of the parent is copied to the new individual. Okay, and for the production of roses, jasmine, orange, banana, such kind of seedless plants that also can be propagated vegetatively. Okay, so this is about vegetative propagation. And have you seen small buds that are arising from the potatoes? Yes, that is also a form of vegetative propagation in potato by bud formation. But small buds are arising and that bud will have the ability to give rise to a new plant. And in bryophyllum, it is a plant that have a vegetative propagation. Their leaves are vegetatively propagated. Their leaves that contains small buds on the notches along their margins of leaves, and that small buds will fall off to the soil and that have the ability to give rise to a new plant. So the leaves are the part which is producing a bud and that will give rise to a new plant. So the plant part that can be used for the propagation of propagation is called as vegetative propagule of that plant. Next, we can discuss about tissue culture. In tissue culture, we know that numerous plantlets can be produced by tissue culture. So for tissue culture, a growing tip of a plant can be selected and it is separated. Then it is transferred to a medium that is artificially prepared. The growing tip of a plantlet is separated and it is transferred to a medium that contains artificial nutrients. So when it is transferred to that medium, it will start dividing and it will produce numerous cells or mass of cells. And that mass of cells are called as callus. So from that callus, it is separated. Callus is separated and it is transferred to another medium that contain growth hormones, plant hormones, and that will lead to the differentiation of the callus cells. Okay, so from the callus cells, new plantlets will arise. And then the plantlets are taken and it is transferred to the soil and where there is growth of that plantlet occur. This is the process of tissue culture, and in the most important thing about tissue culture is it is a setup of a sterile environment that is artificially creating. Sterile environment means the disease free and all the available all the proper nutrients, then proper sunlight, all the requirements are available in that artificial sector. So the plant is protected and the plant is grown having all the availability, and the healthy plantlets, more number of healthy plantlets can be created by tissue culture. Okay, so the commercial production of plantlets are done with the help of this tissue culture. And the next topic that we have discussed was spore formation. Spore formation. It is also an asexual method which is mainly occur in bread mold. How the bread molds are formed? Bread molds are the fungus-like organism. They are formed when the bread is kept in cool, moist and dark places. So, bread mold is otherwise called as rhizopus. Rhizopus. And the body of bread mold is called as hyphae. Hyphae, and that hyphae contains a tiny blob on a stick-like structure. And the plant body hyphae is not the reproductive part of the bread mold. A tiny blob on a stick-like structure is present on their body, and that blob contains sporangia. Sporangia, and this sporangia contains numerous spores. Sporangia contains numerous spore in it, and each spore will give rise to a new hyphae or a new bread mold. I will repeat once more. The tiny blob on a stick on the body of hyphae on the body of bread mold contains a sporangia. And that sporangia contains numerous spores, and each spore will give rise to a new rhizopus. That is the mode of reproduction in asexual reproduction in bread mold. Okay, so till now we have discussed the different types of asexual reproduction, and in that only a single parent is involved, right? So that is asexual reproduction. So the exact copies of the parent plants are created by asexual reproduction. And in our next class, we will discuss the sexual reproduction. What is sexual reproduction? And that is a wide area that we will discuss in our coming classes. Okay, so this is enough for today. Thank you.